Hello. Welcome to today's session of Plant Physiology and Metabolism course. In my previous lecture, we have gone through in detail the process of glycolysis, glycolytic pathway, and we have discussed in detail each steps. Now, we had talked about regarding the switches that occurs in the glycolytic pathway. So now question arises how this glycolytic pathway could be regulated, and this is very crucial for maintaining cellular homeostasis. So you see that the regulation of glycolysis in plant is basically a very dynamic process, and that responds to the cellular energy state and the availability of substrate. And you see that in our previous lecture we have gone into detail. Just I mentioned that the reactions catalyzed by enzymes, particularly hexokinase, phosphofructokinase and pyruvate kinase, are basically exothermic, and these steps display a large change in free energy within existing cellular conditions. So these reactions basically serves as a key point for regulating glycolysis. Once these enzymes are active, glycolysis generally proceeds in forward direction, and glucose is readily metabolized to generate pyruvate. But contrary to this, inhibition of these enzymes inhibit the glycolytic pathway. So these regulatory mechanisms basically help to maintain energy homeostasis and adapt glycolytic pathway to specific needs of the plant cell. So in both plants and animals, key steps in the glycolysis is the conversion of glucose 6-phosphate to fructose 6-phosphate. You see in the glycolytic pathway reaction that conversion of glucose 6-phosphate to fructose 1,6 bisphosphate is very crucial. This step is controlled by two main enzymes, ATP dependent phosphofructokinase, that is PFK, and fructose 1,6 bisphosphate, phosphatase. Plants generally have a unique way of regulating glycolysis called bottom-up regulation, which focuses on the role of pyruvate kinase. Basically in plants, you see that pyruvate kinase is basically influenced by allosteric inhibition from pyruvate and ATP. This regulation basically extends to the control of phosphoenol pyruvate accumulation, becoming a crucial control point in plant glycolysis. So you see in the slide, pyruvate kinase activity in plants is generally inhibited by certain compounds from the tricarboxylation cycle that we will learn in our coming lectures. This complex regulatory network in plant is, you see, serves as a vital for balancing cellular respiration and the production of sucrose or polysaccharides, highlighting the importance of glycolysis in plant metabolism. Another enzyme, hexokinase activity, is basically inhibited by its product glucose 6-phosphate. This is the first step of glycolysis, serving as a negative feedback mechanism. This basically helps to prevent unnecessary phosphorylation when glucose 6-phosphate levels are high. Then another enzyme, phosphofructokinase, that is PFK, plays a very crucial role as regulatory enzyme in the glycolytic pathway, a crucial metabolic process that breaks down glucose to generate energy. Specifically, phosphofructokinase catalyzes the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. You see in the glycolytic pathway, third step basically. And this consumes ATP in this particular reaction, and more importantly, phosphofructokinase exhibits also allosteric inhibition by high concentration of ATP, serving as a sort of sophisticated cellular feedback mechanism. This basically inhibition signifies that when cellular energy levels are potential, plentiful and ATP is in abundant, generally phosphofructokinase activity is reduced, effectively slowing down the process of glycolysis. In addition to this, phosphofructokinase sensitive to another key metabolic signals, that is citrate, and citrate is an intermediate of the citric acid cycle. The inhibition of phosphofructokinase by citrate further underscores the ability of the cell to modulate glycolysis in response to the overall energy status. When citrate levels are elevated, which is indicative of ample energy in the form of citric acid cycle intermediates, generally phosphofructokinase is inhibited, contributing to the down regulation of the glycolysis. You see, it's very much clear in the pathway that have been shown with red arrow. This allosteric regulations highlight the intricate control mechanism that cells generally employ to fine tune their metabolic activities based on availability of energy resources. Then another enzyme you see in the pathway is pyruvate kinase. Basically, this pyruvate kinase plays a very crucial role in glycolysis. The metabolic pathway responsible for converting glucose into pyruvate, and basically this enzyme pyruvate kinase catalyzes the conversion of phosphoenol pyruvate to pyruvate, and that results in the production of ATP. And in addition to this, pyruvate kinase is subject to dual regulation to maintain a delicate balance in cellular energy metabolism. On one hand, then this enzyme is inhibited by ATP. Pyruvate kinase is inhibited by ATP, which is the end product of glycolysis, ensuring that when cellular ATP levels are high, glycolysis is slowed down to prevent unnecessary ATP production. On the other hand, pyruvate kinase is basically activated by fructose 1,6 bisphosphate, a glycolytic intermediate. So, this activation occurs when glycolytic intermediates are present in abundance, signaling that there is a sufficient supply of substrate for the process of glycolysis. The intricate regulation of pyruvate kinase thus acts as a sort of molecular switch. We have discussed in earlier that this pyruvate kinase basically acts as a molecular switch for fine tuning glycolytic activity based on cellular energy status and availability of glycolytic intermediates. In addition to this, allosteric regulation plays a crucial role in the control of enzyme activity involving the binding of molecules at sites distinct from the active sites of enzyme to influence their function. In context of glycolysis, you see that a central metabolic pathway, several enzymes are subject to allosteric regulation by metabolites such as adenosine monophosphate, adenosine diphosphate and adenosine triphosphate. These molecules serve as key indicators of cellular energy status. The presence of adenosine monophosphate and ADP typically signifies a low energy state within the cell, prompting or invoking allosteric activation of glycolytic enzymes to accelerate the production of ATP through glycolysis. Conversely, you see that an abundance of ATP signals a high energy state leading to allosteric inhibition of glycolytic enzymes to prevent unnecessary production of ATP. This allosteric regulation ensures that cellular energy production aligns with the dynamic energy demands of the cell, thereby contributing to overall homeostasis of energy metabolism. So, you see that these switches basically maintain a proper homeostasis. Like in electrical circuits, you see different switches as sort of control point. You just switch on, then bulbs uh, illuminate, and different switches serve different functions. So likewise in the glycolytic pathway also, basically these control points serve as a sort of switch to regulate metabolism taking place inside the cell, and it is on and off as per the demand of the cell. So it's very interesting to share these facts. Uh, you should enjoy that how glycolytic pathway are basically regulated. Now, once pyruvate is formed, pyruvate metabolism is a very essential biochemical process that is central to generating cellular energy. Basically, it converts pyruvate, which is a 3-carbon compound you might have seen, into various metabolic pathway associated with the carboxylic acid cycle. The tricarboxylic acid cycle, which is a cyclic pathway crucial for cellular respiration, serves as a hub for oxidizing different carbon compounds including pyruvate. So basically pyruvate enters the TCA cycle through a series of reactions, ultimately transforming it into carbon dioxide and high energy electron carriers like NADH and FADH2. What happens generally in aerobic conditions, pyruvate, the end product of glycolysis, undergoes oxidation inside the mitochondria to form acetyl coenzyme A. This is basically happens in the case of in aerobic condition. This acetyl coenzyme A then participate in the citric acid cycle. Simultaneously, NADH is produced during NADH produced during glycolysis oxidizes to NAD through the NADH shuttle system. It's important to note here that specific transports are lacking for direct transport of NADH across the inner mitochondrial membrane. On the other hand, you see that under anaerobic conditions, the pyruvate follows a different metabolic pathway and undergoes fermentation. Now, this is the whole story in nutshell, that pyruvate, what happens under aerobic conditions and what happens in anaerobic conditions, leading to distinct outcomes under both these conditions. Now, what happens when oxygen is present? That is aerobic condition. Generally, in this condition, pyruvate is transported into the mitochondria. Then within the mitochondria, pyruvate undergoes oxidative decarboxylation, a process that converts it into acetyl coenzyme A. Acetyl coenzyme A then enters to the citric acid cycle, also referred to as Krebs cycle, where it participates in a series of reactions that produce NADH and FADH2. These molecules carry high energy electrons. Additionally, you see that NADH generated during glycolysis is oxidized to NAD within the mitochondria through the electron transport chain. This process leads to generation of ATP through oxidative phosphorylation. Now, what happens under anaerobic conditions? Under anaerobic conditions, or you can say where there is a limited access of oxygen, pyruvate has to undergo fermentation. Different organisms and metabolic pathways can lead to the conversion of pyruvate into various end products such as lactic acid or ethanol through the process of fermentation. Basically, this fermentation serves the purpose of regenerating NAD and from NADH, which is crucial for sustaining glycolysis in the absence of oxygen. NAD is an essential cofactor for the glycolysis, and the accumulation of NADH can impede or halt the process of glycolysis. So, this is also very crucial. Now, what is the fate of pyruvate once it reaches into the mitochondria? When oxygen is present, or you can say under aerobic condition, pyruvate is carried into the mitochondria. Inside the mitochondria, pyruvate undergoes oxidative decarboxylation, which converts it into acetyl coenzyme A. Acetyl coenzyme A basically then enters the citric acid cycle, also known as Krebs cycle, where it participates in a series of reactions that produce NADH and FADH2, which carry basically high energy electrons. And NADH generated during glycolysis is also converted back to NAD in the mitochondria through the electron transport chain, resulting in the production of ATP through oxidative phosphorylation. Now, this basically oxidative phosphorylation of pyruvate, which serves as a sort of link reaction, and then basically in the inner mitochondrial membrane, this inner mitochondrial membrane basically serves as a sort of barrier and require the assistance of various transporters for the passage of molecules. Basically, the inner mitochondrial membrane is, you see, that intricately folded to form cristae and encompasses approximately 50% of the total mitochondrial proteins. Then different mitochondrial compartments also serves as a very crucial role. That you see that the two membranes of the mitochondria enclose a compartment known as matrix, and nearly 50% of the mitochondrial molecules by weight are found in the matrix. The matrix has very limited water content, potentially leading to the organization of matrix protein into multi-protein complex called metabolons. Then pyruvate basically, which enters into the mitochondria, undergoes oxidative decarboxylation to form acetyl coenzyme A. During this conversion, one carbon dioxide is lost as carbon dioxide, and the process is basically catalyzed by pyruvate dehydrogenase complex. Then inside the mitochondria, oxidation and NADH production takes place. The oxidation of pyruvate is basically coupled with the reduction of NAD to NADH. This key biochemical reactions is basically facilitated by the pyruvate dehydrogenase complex. Then what is the role of this link in plants? In plant, basically pyruvate dehydrogenase complex is not exclusive to mitochondria, it is also present in plastids. In plastid, pyruvate dehydrogenase complex participates in lipid biosynthesis, showing its versatility in different cellular compartments. The conversion of pyruvate basically to acetyl coenzyme A, facilitated by pyruvate decarboxylase, is referred to as link reaction, and this link reaction basically bridges glycolysis and tricarboxylic acid cycle, connecting these crucial metabolic pathway inside the cell. Now, in the first step, basically pyruvate undergoes decarboxylation by reacting with activated pyrophosphate of the enzyme pyruvate dehydrogenase, resulting in the formation of hydroxyethyl derivatives. So, this is the first step. And then pyruvate dehydrogenase also facilitates the second step, where 2 electrons and the acyl group are transferred from this TPP to the oxidized form of the lipoacyl group of the coenzyme, that is dihydrolipoyl transacetylase, that is E2, leading to the creation of acyl thioester of the reduced lipoyl group. This third step involves a trans-esterification process in which the thiol group of coenzyme A replaces the thiol group of second complex, that is E2, and produce acyl coenzyme A and the fully reduced form of the lipoyl group. In the fourth step, dihydrolipoyl dehydrogenase, that is E3, facilitate the transfer of 2 hydrogen atoms from the reduced lipoyl groups of E2 to FAD prosthetic group of E3, restoring the oxidized form of lipoacyl group of E2. Finally, in the fifth step, the reduced FADH2 of E3 transfers a hydride ion to NAD1, resulting in the formation of NADH. At this point, the enzyme complex is ready for another catalytic cycle. So, composition of enzyme 1 tetramer, basically E1 is a tetraheteromer comprising of alpha and beta subunit, covalently linked with thiamine pyrophosphate, that is abbreviated as TPP. So, basically this first enzyme complex E1 is covalently linked to TPP. Now, how the activity of E1 is regulated? Basically, phosphorylation of 2 conserved serine residue of E1 in the alpha subunit leads to inactivation. Then further dephosphorylation of these serine residue converts E1 into an active form. Then how E1 subunits are connected? E1 subunit is connected to E2 core. Basically, E1 subunits are covalently linked to the E2 core, and E3 dimer and binding sites. Basically, E3 exists as a dimer with binding sites for flavin adenine dinucleotide, that is FAD, and nicotine amide adenine dinucleotide, that is NAD. And these are linked to the E2 core on the inner side of the complex. You see that pyruvate dehydrogenase complex and this phosphopyruvate, basically these enzymes are very sequentially regulated, and this pyruvate dehydrogenase kinase catalyzes ATP dependent phosphorylation of E1 complex, leading to its inactivation. Then phosphopyruvate dehydrogenase phosphatase, that is PDP, catalyzes the dephosphorylation of E1, resulting in the activation. Now, what is the fundamental role of pyruvate dehydrogenase complex and phosphopyruvate dehydrogenase complex? Basically, this pyruvate dehydrogenase complex is responsible for inactivation of the enzyme complex, and phosphopyruvate dehydrogenase phosphatase PDP is involved in the activation of the enzyme complex. So, it could be summarized that E1 basically tetraheteromer complex is intricately regulated through the phosphorylation and dephosphorylation events involving the E1, E2 and E3 subunits, as well as regulatory enzymes PDK and PDP. This tight control ensures the sort of dynamic modulation of enzyme activity in response to the cellular needs. So this is the sketch of how different groups attach phosphorylated and dephosphorylated, and it's a sort of visual representation of PDH enzyme, including its different subunits and cofactor and how it functions, as I have discussed in detail in my previous slide. Now, how the oxidative decarboxylation of pyruvates occurs. Basically, this oxidative decarboxylation of pyruvate occurs in basically 5 steps. So, we will go into the details how these steps are regulated and how these steps could lead to formation of different intermediates. So, first step basically is pyruvate dehydrogenase complex, that is E1-TPP. This is an enzyme that relies on thiamine pyrophosphate and is involved in the decarboxylation of pyruvate, producing a hydroxyethyl TPP intermediate. So, you see in the slide the first reaction. This process is equivalent to the one carried out by yeast pyruvate decarboxylase. Then second, unlike pyruvate decarboxylase, pyruvate dehydrogenase does not convert hydroxyethyl TPP into acetaldehyde and TPP. Instead, it transfers the hydroxyethyl group to the lipoic acid of the subsequent enzyme dihydrolipoyl trans acetylase E2 in the complex, creating a thiester bond. This is very crucial. So, this reaction is linked to the removal of TPP and the creation of acetyl dihydrolipoamide accompanied by simultaneous release of active E1. The hydroxyethyl is then oxidized to an acetyl group through the concurrent reduction of the lipoamide disulfide bond. Then you see in the slide, E2 basically catalyzes the transfer of acetyl group from acetyl dihydrolipoamide to thiol coenzyme A, yielding acetyl coenzyme A and dihydrolipoamide E2. You see in the reaction. Then the fourth, dihydrolipoyl dehydrogenase is also known as E3 or dihydrolipoamide dehydrogenase, plays a very crucial role in the enzymatic cycle of E2 by reoxidizing dihydrolipoamide. Basically, this process involves the oxidation of dihydrolipoamide coupled with the reduction of covalently attached cofactors FAD and FSDS2 and the formation of lipoamide disulfide bond, that is SS bond. Then reduced FADS2 could be reoxidized by the simultaneous reduction of NAD to NADH. E3 has got the binding site for both FAD and NAD. As a result, the sequence of these 5 reactions, the net result of oxidative decarboxylation of pyruvate is the formation of acetyl coenzyme A, NADH and loss of carbon as carbon dioxide. So, how the activity of pyruvate dehydrogenase complex is regulated? This is also very crucial. Regulation of the activity of pyruvate dehydrogenase complex, or it is abbreviated as PDH. So, pyruvate dehydrogenase initiate a series of crucial reactions, is basically tightly regulated to control the cellular metabolism. And you see that how the activity of this enzyme is regulated, and basically the ratio of ATP and ADP, which is reflective of the metabolic state of the cell, plays a very crucial role in the regulation of pyruvate dehydrogenase complex. So this is serves as a sort of indicator. Then E1 is basically inactivated by ATP dependent phosphorylation. Pyruvate dehydrogenase kinase basically catalyzes the process of phosphorylation. A phosphatase is basically responsible for removing the phosphoryl group from E1, restoring its activity. Pyruvate dehydrogenase activity is inhibited by end product of the reaction, that is acetyl coenzyme A and NADH. Basically, pyruvate acts as an activator by inhibiting PDH kinase activity. This inhibition in turn removes the phosphorylation based inhibition on the enzyme. Ammonium ion activate the PDH kinase activity, leading to the inhibition of PDH activity. So basically, the interplay of ATP and ADP ratio, phosphorylation and various activators and inhibitors creates a regulatory system for pyruvate dehydrogenase complex, ensuring cellular energy balance inside the cell. So thank you very much for today's session. We have learned how glycolytic pathways is regulated, and then how pyruvate is converted into acetyl coenzyme A, how various complex regulate this particular step inside the mitochondria. Thank you very much.